Exactly. Yeah. So I mean, it was a big coincidence with Mikhail's work and Sharanfu's. I'm glad I'm here. I'm glad we are talking to each other. Um, so thanks for that. I'm going to be talking about our work that has started quite many years ago, and the COVID kind of like was in between. So we really care about genetic variants and then how 3D organization helps them find their targets, and more importantly, today how genetic variants may impact those chromatin organization. So good news and bad news. We are taking a little bit of a break. We do love deep learning and we use it in other projects, but at least for today, for this 20 minutes, no deep learning. So enjoy that. I'm going to do something very quick in the first 10 slides or so. So 3D genome matters. We really like it. Hi-C is an amazing method. We've been using it. A lot of other people using it. Great data generated from it. Provided us a lot of insights and a lot of tool development opportunities, all the way from the compartments to TADs to loops. And the other thing that it has helped, and this has been mentioned, that we all are trying to figure out what the heck these non-coding variants are doing when they're especially associated with the diseases and phenotypes. So Hi-C and its variants have helped us actually create a bit of a resource where we were able to map in primary human immune cell types. And again, that's another work that's parallel to what Mikhail has done many years ago, where we looked at 5 different immune cell types, used a variant of Hi-C that's called Hi-CHIP, and then mapped chromatin interactions for those immune cell types, where we also had a matching cohort of EQTLs, as Mikhail mentioned, as expression quantitative trait loci. We had about 100 donors. The Hi-CHIP data we had from 6 donors only because it's expensive. Back then, when we generated this data, people were mainly using loop callers from Hi-C and other domains for Hi-CHIP data. We developed one that we really like and has been highly used to identify interactions such as those you're seeing that links ideally promoters to validated enhancers without too many false positive interactions. That was work by Soria, and then actually pretty much all the work that I'm going to talk about today is also led by Soria, who's a longtime postdoc of mine, very talented computer science PhD student. So what we did with the resource, with EQTLs and with the chromatin contact maps we generated using HTK27 acetylation Hi-CHIP data, was just to take a real look at how we do the EQTL mapping, right? So if you looked at GTEx and other cohorts, generally you take a gene promoter or the PSS, look at 1 megabase around it, plus minus, and just ask the question, every single SNP here, do they correlate with gene expression, right? So the idea is why test everything, right? I mean, a lot of those SNPs are probably not overlapping anything of significance in terms of regulatory activity and so on and so forth. And the other part of it is that why not test things that are further away if they give you a clue that they might matter, right? Such as a very long-range chromatin. So we did that, and then we have published this. I'm not going to go into details of it, but that gave us a lot of interesting insights about cell type specificity of EQTLs, these cryptic things where it's an EQTL in one cell type, but not in the other one, or it's in the different directions. We have identified promoters that themselves don't drive gene expression of the canonical gene, but do serve as enhancers through looping to other genes, which is something I think that's more and more becoming commonly appreciated, that both being a promoter and an enhancer is a kind of like a spectrum, right? Enhancers transcribe, promoters to regulate distally. And then it allowed us what we really wanted, gave us enough many statistical tests that we save from that within 1 megabase, so that we ask questions beyond 1 megabase and identified a couple of validated, CRISPR validated enhancers that are beyond the 1 megabase range. So that's one issue, right? Like the non-coding regions and how you link them to gene expression or anything in GWAS. the other issue is that there's still a lot of missing variability, that we are unable to explain a large proportion of variation for both traits such as height and diseases just by looking at genetic variants. And EQTLs are trying to help there, but EQTLs are also, as just recently been postulated by Jonathan Pritchard's group, they are systematically different, right? The GWAS SNPs are those SNPs that are like distal to the genes. EQTLs are closer to the promoters. They have different selective pressures on them and so on and so forth. So EQTLs are not necessarily completely explaining or capturing, and they have a limited overlap with the GWAS. So there's still that missing part, which we hope that the other QTL studies, such as one you heard today, metal QTL and chromatin QTLs, open accessibility QTLs, and so on and so forth, may help bridge. So what we did is we asked a question similar to morning's talk, whether there are SNPs, there are non-coding variants that are associated with the looping strength, right? Just like an EQTL, but now we are looking at the loop strength. And for this here, we are using, the differences is that we are focusing on CD4 naive human T cells here, which is our favorite. And also we are trying to do this genome-wide without focusing on a predefined set of regions. So we are using H3K27 acetylation high chip with the hope that it covers pretty much all active regulatory elements, including active enhancers and promoters and so on and so forth. So this didn't came out of the blue, right? We've been thinking about this for many years by now. We, when I first joined my group or the institute, there was an ongoing project where actually this concept emerged and got validated, where we looked at a specific locus that is associated with asthma and a whole bunch of autoimmune diseases. And then we were trying to narrow down which SNPs might be causal for the changes in gene expression that are likely causal for the disease association or the disease risk differences. And as you can see here, there are two different regions. This is a 300 kb or so haplotype with the high LD. So pretty much everything is in LD with each other. So here, this risk allele here goes together with this one. They are the risk haplotype, and then this non-risk allele goes together with that one. And then when we looked at these in more detail using ChIP-qPCR and looked at the CTCF binding, we were able to see that actually there is a switching of CTCF binding, right? So in risk allele, CTCF is binding here, whereas in non-risk allele, CTCF is binding more here. So there's a reorganization of chromatin configuration. And then we validated that using 4C at the time. Here you can see that the 4 donors with the homozygous reference, 4 donors with the homozygous alternative allele, and then the chromatin reorganization for the specific gene where we designed the bait, that suggests us that in the non-risk allele, you don't have access to these distal elements, distal regulatory regions, arguably promoter and enhancer at the same time, whereas in the risk allele, you do have a strong loop there, and then that, explains the overexpression of the gene. So that motivated us, and then the availability of the EQTL cohort that we have, which we call DICE. So we focused on one cell type, used 30 donors. We have ChIP-seq data, RNA-seq data, and then high-chip data using H3K27 acetylation. For some of them, we have attack-seq data too. So we developed a pipeline to do this properly. So that involves peak calling from ChIP-seq, using those peaks with our FitHiChip tool to call loops, and also using genetic variants to then drive associations. And here, as you can see, the idea is to be able to look at genotype-dependent trends as well as allele-specific trends and combine them, filter them, and overlap them with a myriad of different data sets and different signatures, such as transcription factor binding sites, remap, as mentioned, Adastra, which gives you allele-specific imbalance, FineMap, GWAS SNP, co-localized SNPs, and so on and so forth. At the end of the day, we ended for these 30 donors, we ended up calling about 266,000 ChIP-seq peaks, and then they were used to call about 600,000 regulatory loops at 5 kb resolution. And then after that, for each loop, we looked at each anchor plus minus 1 bin of each anchor at 5 kb and asked whether the SNPs in those anchors are associated with the looping. So when we did that, we found about 15,000 SNP loop pairs. Right? If you know the EQTL literature, I think you will know that there are pairs and there's also SNPs, there's also loops. So we have about 9,000 SNPs that are connecting or associated with about 2,300 different loops among all these 600,000 loops that have a statistically significant association and survived a whole bunch of filterings that we have applied. When we looked at these loops, they actually mainly were coming from 2 or more donors out of 30. So some were significant in everything, but their strength was changing, whereas some loops were significant in a subset of donors and not in the other. And then these loops were stronger compared to overall background loops that we have called. When we looked at their overlap with EQTLs, there was a good subset of them that overlap with non-EQTLs from these 2 different cohorts. DICE is the one we have, and Immunex UT is from Japan. Different populations, different cell types that they have looked. YY1, CTCF, SP1, non-looping factors, as well as things such as IRFs that are interferon response factors, transcription factors that are able to modify chromatin in the immune cell context came up for these IQTL regions. When we looked at their enrichment for some diseases, we were able to see that the IQTLs that we have were more enriched for disease-associated SNPs than the overall EQTLs. And we did the stratified LD analyses that also LD correlation analysis that also confirmed that. When we went back to the specific example, the asthma locus that we have, what we see is a specific loop QTL there that changes the looping by about 60% or so. And then when you look at those SNPs, you can see that they are also EQTLs, right? Remember that this whole haplotype is like a big chunk. So what one SNP is correlated, the other ones are going to be correlated too. So that confirms that dependence of looping to genotype in that specific locus. And then if we went and looked at another important region, which is the T-cell receptor zeta chain, we found that there is a pretty strong, effect size loop QTL there with, as you can see, a pretty nice trend across different donors in terms of how strong the looping is. And this, likely associates with the CD247's expression. When we looked at the specific set of SNPs, you can see that from the ADASTRA database, you can see that one of them do create an imbalance in ETS1 binding, so allele-specific imbalance, whereas another one hits a very strong IRF1 motif. So suggesting that an IRF1, as I mentioned, can remodel the chromatin, so does ETS1. So suggesting that the change of looping may be associated with these specific transcription factors. And then when we looked at this further, what we have seen is that although we are looking at naive CD4 cells, the effect size of EQTL is much higher in other CD4 subsets, more memory and differentiated subsets, such as Tregs here. So that gave us an idea as to maybe by looking at a cell type, such as naive CD4 cells, we may infer things about what may happen later, right? So there might be IQTLs in the cell type that you're looking at that are not EQTLs yet, but upon stimulation, differentiation, perturbation, they may become EQTLs in the next stage. So we did find such examples, and such examples have been called dynamic EQTLs in the literature. And as you can see in maybe CD4 naive cells, you have a flat expression pattern, whereas you have a very strong expression dependence on genotype. We have found about 500 such loops, and then 1,200 such SNPs, and 1/3 of them overlap with regulatory peaks. So we have some other examples of it, and then we have examples where we have IQTLs that are not EQTLs in any CD4 T cell subset, so likely suggesting that they may have different mechanisms. So one interesting concept that came out of this also was the connectivity QTLs, right? So when we looked at these IQTL SNPs, some of them associated with many different loops and the whole connectivity. This is one example. So this was IQTL for all these loops, but as you can see, it is also for the connectivity of the whole region. Very strong pattern dependence on genotype, and then that's reproduced when you aggregate these scores. Some more examples that I don't have time to go into, but at the end of the day, I think we have defined interaction QTLs in CD4 naive cells. We have found some that are dynamic, that are going to be EQTLs later down if the CD4 cells differentiate in the memory subsets, and also SNPs that are associated with the overall connectivity of the region rather than one loop at a time. So with that, I'd like to thank my lab and my collaborators, and I'll take any questions.
Thank you. Is there any question to start with? Otherwise, I will start with my question that I had about this, specifically about this dynamic EQTLs. So how does that work in your mind mechanistically? Like, what would be the idea here?
So think about in a specific state of cell that looping is already established, right? There is a pre-configured chromatin state, and the right transcription factors may or may not have been there, right? So once you are differentiated into the next stage, the transcription factor landscape changes, and that likely, and the expression of the gene changes too. Sometimes it is induction of the gene expression, and sometimes in the one that I show, the gene expression is still there, but at lower levels in one state, it kind of like increases in the next state, and then now you see the separation of genotypes. So that's a concept that's been coined and has been out there, the dynamic EQTL concept. So I guess our contribution is that we are showing that some of them are loop QTLs in a state that you can look at, with implications to things such as like our limited ability to look at specific cell types and not all their perturbation or stimulation states. So maybe you can look at one baseline state and then infer molecular QTLs such as loop QTLs, and that may give you an idea for future configurations.
So you've shown a couple of nice examples of conserved TF motifs, but do you also have like a lack of a common theme for like one or two transcription factors that are there? Is it also diverse in the range?
Yeah, I mean, I have shown, I think, the motif enrichment analysis that's coming up with the, in the broader regions, that's coming up with some certain common factors. But in terms of the examples that we have derived, there is a lack of it. So it's sometimes CTCF, but sometimes no CTCF is there to be found, and there's this immune cell related factor. So it's not one size fits all, just like yours, I think. And also, there are a couple of these examples where we find the same SNP or the same set of SNPs perturb different transcription factor binding sites. They're in high LD, so like it's exciting, but it's very difficult to explain. Some are activators, some are repressors, and so on and so forth.
Or even the same SNP sometimes.
Yeah, even the same SNP sometimes overlaps two different motifs. Yeah. Good that we have some consistency. Research is reproducible sometimes.
Yeah.
So since we have 1 minute left, I'm going to ask a follow-up question to the one I had before. So these loop QTLs, do they then go in the same direction of the dynamic EQTLs later on? So have you checked the directionality there?
Yeah, so there is, in general, yes, but there have been cases, just like what Mikhail has shown, that we believe some insulatory mechanisms are in place, that the loop goes higher with a specific genotype.
How many of these cases do you have?
We haven't quantified. It is kind of difficult to quantify, so we haven't quantified in terms of the directionality in the dynamic QTL context.
And then can you use your loop QTLs to actually predict which QTL, variant would be a dynamic EQTL maybe in other conditions that have not been measured yet? Would you go as far?
Yeah, I mean, we would need validation of some sort, so we would still need EQTL data. So here we look at CD4 naive cells, and then between those two different EQTL databases, we have about like 8 or so different CD4 T cell subsets. So that's our validation that some of them go into that, but we haven't done in an unseen cell state, for instance.
Okay, then there's one last question from the online participants. How are you doing the fine mapping of GWAS?
We use fine map GWAS SNPs from CausalDB directly. I mean, but when we did it ourselves manually in the previous iterations of the paper, we used fine map, the fine map package from GWAS data.